Hello everyone, it's Nick Dalkis, a student of biochemistry and biotechnology, and I'm glad to host today's podcast about NITRiC. NITRiC is a research project hosted by the Lab of Plant and Environmental Biotechnology at University of Thessaly and funded by the Hellenic Foundation for Research and Innovation and the General Secretariat for Research and Technology. With us today is the PI of NITRiC, Evangelia Papadopoulou, who is also a postdoctoral researcher in environmental microbiology at University of Thessaly. How are you doing, Evangelia?
Hi, Nikos. Thank you for the introduction. I'm really glad to discuss with you.
Okay, so I'll start the conversation with some information I gathered regarding your work and your field, and then you can hopefully fill in the details. So modern agricultural systems depend heavily on large inputs of synthetic nitrogen fertilizers to maintain crop productivity and meet the increasing global food demands. However, approximately 70% of the annual global input of nitrogen fertilizer is lost from agricultural ecosystems due to a series of microbially mediated processes related to the nitrogen cycling, increasing significantly the farmers' costs. To date, several technologies controlling processes in nitrogen cycling have been developed to help in reducing losses of nitrogen in soil that would otherwise be used by crops. Nitrification inhibitors constitute one of these technologies, which is actually the main focus of your project. So, Evangelia, can you explain us what are exactly these compounds and how do they work?
Well, nitrification inhibitors are chemical compounds that are routinely incorporated into nitrogen-stabilized fertilizers to slow down nitrification, the biological oxidation of ammonia, ammonium-containing salts or urea, which are applied to soil as fertilizers, to nitrate. Actually, they reduce the activity of the microorganisms participating in nitrification, mainly by interfering with the key enzymes involved in the oxidation of ammonia. This way, they extend the presence of the more efficient in terms of plant productivity form of ammonium in the rooting zone, which could improve the match between available nitrogen supply and plant demand, leading thus to enhanced crop productivity.
The environmental concerns of nitrification are also contribute to interest in the use of nitrification inhibitors. The primary product of this process, nitrate, leaches into groundwater, if I'm correct, producing acute toxicity in multiple species of wildlife and contributing to the eutrophication of standing water.
This is absolutely true. The widespread synthetic fertilizer usage in the 20th century, apart from ensuring food safety for the increasing global population, have pushed the nitrogen cycle beyond sustainability with serious risk for the health and the environment. Eutrophication, groundwater and atmosphere pollution, biodiversity loss, and climate change are some of the most pernicious global environmental challenges we currently face and are all directly linked to nitrification processes in fertilized soils. Nitrification generates the soil-mobile anion of nitrate from the relatively immobile ammonium pools, causing significant losses of nitrogen from agricultural systems via nitrate leaching and providing substrates for both nitrifiers and denitrifiers to produce toxic, volatile derivatives like nitrous oxide, a greenhouse gas with 300 times the heating trapping capacity of carbon dioxide per molecule, and the dominant ozone-destroying agent of the present century.
Although nitrification inhibitors are usually effective in restraining those negative environmental effects and increasing recovery of nitrogen fertilizer in row crops, the level of their effectiveness depends on external conditions, and their benefits are most likely to be seen at less than optimal nitrogen rates. Is this right?
Yes, that's right. The bioactivity of nitrification inhibitors in soil is influenced by various edaphic, agronomic, environmental, and microbial factors that affect not only their performance and their persistence in the agricultural ecosystems, but also the activity of the microorganisms contributing to ammonia oxidation in agricultural soils. Data coming from several studies have indicated that both relative abundances and activities of soil nitrifiers vary with soil type, climate, vegetation, and other factors like soil pH, temperature, soil moisture content, source of nitrogen used for fertilization, and etc.
So, nitrification inhibitors seem to be potent agents in increasing fertilizer efficiencies in the field, but is there still actual room for improvement?
Definitely, yes. All currently known nitrification inhibitors were developed in the 20th century, while our understanding of the microbiology and biochemistry of nitrification is continuously changing in fundamental ways. Relatively recent discoveries, impelled by the technical breakthroughs of metagenomics, have revealed new types of organisms and processes, changing thus profoundly the way we view and study reactive nitrogen transformations in soil and the factors that control them.
Well, in nitrification, the biological oxidation of ammonia or ammonium to nitrate, was long considered as a two-step process carried out by ammonia oxidizing bacteria and nitrite oxidizing bacteria. Ammonia oxidizing bacteria oxidize ammonia to hydroxylamine, which is further oxidized to nitrite, and the nitrite oxidizing bacteria, in their part, transform subsequently nitrite to nitrate. So, what's new about this microbially mediated process?
Well, indeed, this is the traditional perspective of soil nitrification. However, over the last 15 years, other groups contributing to soil nitrification were discovered, like ammonia oxidizing archaea and recently Comamox nitrospira, which are able to perform complete oxidation of ammonia to nitrate within an individual cell. These organisms are all known to contribute to ammonia oxidation in agricultural soils, but it is difficult to estimate their relative contribution as different environmental factors can affect their abundance and activity.
And what about the activity spectrum of the currently available for use in agriculture nitrification inhibitors? I mean, are these compounds equally effective against all known microbial nitrification
Well,
contributors?
this is in question. The majority of the currently available for use in agriculture nitrification inhibitors has shown a selective inhibitory activity towards ammonia oxidizing bacteria, while their impact on ammonia oxidizing archaea is either limited or unknown. In addition, no information is available regarding the impact of nitrification inhibitors on other functional microbial groups like nitrate oxidizing bacteria or Comamox bacteria.
So, based on these observations, there is actual need for further evaluation of the activity range of the agriculturally relevant nitrification inhibitors.
Indeed, most of these data have been derived from soil microcosm studies, where ammonia oxidizing bacteria were functionally dominant, and only a few studies evaluated the activity of nitrification inhibitors of agricultural relevance on ammonia oxidizing archaea using pure cultures. Within this frame, the use of inhibition assays with pure cultures of a diverse range of soil-derived strains is definitely necessary to define the exact spectrum of activity of nitrification inhibitors destined for use in agriculture.
This variation in the sensitivity of ammonia oxidizing archaea and bacteria to the different types of nitrification inhibitors and the possible important contribution of ammonia oxidizing archaea and Comamox bacteria to nitrification in distinct ecological niches, as you already mentioned, suggest a suboptimal efficiency for the current available inhibitors and stresses the need for the discovery of novel nitrification inhibitors with a broader range of activity against all microorganisms contributing to nitrification. Uh, but is this actually a realistic scenario?
Well, the development of nitrification inhibitors is indeed both very time consuming and expensive because these products have special characteristics. First of all, they must not have any unfavorable side effects on soil fertility. In addition, they should not degrade to toxic substances in the soil, they should not be toxic to plants, animals, and man. They should fit into the complex production systems of the producers to be economically viable and environmentally acceptable, and they should be stable during production, storage, transport, and use, and their costs should be acceptable to farmers. But apart from the characterized nitrification inhibitors, other agrochemicals can also affect the organisms responsible for nitrification and could, under certain conditions, serve as a source of novel potential nitrification inhibitors. Actually, this is the case of our study. In previous soil microcosm studies, we found that ethoxyquin, an antioxidant used as a preservative in fruit packaging plants, and its derivative, quinonimine, strongly inhibited the activity of both ammonia-oxidizing bacteria and archaea. Ethoxyquin in soil is rapidly transformed to quinonimine and ethoxyquinoline, and this capacity of ethoxyquin to be rapidly transformed in soil to possibly potent nitrification inhibitors is of particular interest, considering that the spectrum and the duration of inhibition are desirable attributes for nitrification inhibitors used in agricultural practice.
So, what are your future research plans?
Through NITRIC, we aim to provide a holistic assessment of the inhibitory effects of ethoxyquin and its oxidation derivatives on nitrification. To achieve this, we will go through a gradual experimental scaling up from culture to soil inhibition assays in combination with advanced molecular and biochemical tools. We will then compare inhibitory effects at all levels and contrast them to that for currently known nitrification inhibitors like dicyandiamide, nitrapyrin, and dimethylpyrazole phosphate. We are also planning to expand our assessments to nitrite oxidizing bacteria and Comomox bacteria and on other microbial groups participating in downstream processes in nitrogen cycling like denitrifiers, for which little is known about their sensitivity to nitrification inhibitors. So, a verification of the universal inhibitory effect of ethoxyquin and quinonimine might lead to the development of a novel universal nitrification inhibitor for more efficient nitrogen conservation in agricultural and pasture soils.
Okay, thank you, Evangelia, and thank you everyone for listening to our podcast session. We would love to hear from you, any thoughts or questions you have, and answer them. Get in touch with us on Twitter at nitric_plantenv, and don't forget to visit our website at nitric.bio.uth.gr. Stay tuned for any further developments. Thank you.